Okay, good morning. It's a very great pleasure for me to give the Pritchard lecture. Can I please have the first slide? It makes life a little bit more easy if you have your slides. Something is happening. I can see it. Yes, thank you very much. Okay, so please note my new email address. I will send my new details to Katie and ask her to distribute it among the Histiocyte Society. I recently moved to Rotterdam to the Erasmus Medical Center. So this is my new email address, and I give this presentation on behalf of the other members of the steering committee. So I'm going to talk about the major topic of the 2022 meeting in last May in Athens. And this is just a nice picture of the harbor where we had one of our dinners. So Carl already showed this slide yesterday, and this is really what is the major theme within the Nikolas Symposium. It is to have together a group of people, experts on a certain topic, and to eat, drink and argue about the topic that the steering committee selects each year for the next meeting. But this year we had something extra because we also honored Dr. Pieter Lenen, who is an immunologist from my country, from the Netherlands. And Pieter Lenen has been working, has been part of the steering committee and has been involved in the Nikolas Symposium for many, many, many years. And now he's close to his retirement, and he has been honored for his service for the Nikolas Symposium and also for his work that he did on monocytes and macrophages. So here I listed some of the previous themes of the last years, and obviously we did not have a meeting during the COVID pandemic, but here you can see that there are many different topics that once every while come and are discussed. So we talk about cellular origins of dendritic cells, we talk about signaling, we talk about the MAP kinase pathways. But once in a while, we also discussed central nervous system inflammation, or for instance, mechanisms and long-term consequences of neuroinflammation. So that's really a recurrent topic, and that obviously has to do with the medical condition of Nikolas Kontoyiannis, the son of Paul and Elizabeth Kontoyiannis, who organize this really fantastic meeting every year. And this year we actually celebrated the 30 years of the Nikolas Symposium, and the major theme of the meeting was neurodegeneration in LCH. Also we discussed some other interesting work, particularly some nice clinical studies that are performed in Greece, but the major theme, and I'm also only discussing the presentations related to the major theme of 2022. So here are some of the cell types that I will come back to during my presentation. So we'll talk about monocytes, macrophages, neurons and microglia cells. So these will be the recurrent themes. And then I composed the presentation as such that I first discuss with you the clinical picture, myeloid cells in the brain, and then lastly therapeutic targeting of cells residing in the brain. And I just want to make an easy disclaimer, I'm not a physician, so there are obviously more experts in the audience that can really talk about the neurodegeneration and the clinical picture. So if I say something wrong, please step up to the microphone and correct me. So these are some of the slides they were kindly provided to me by Matthias Wilk, who was also one of the Pritchard Fellows this year at the meeting. And this is really work that was presented by Eli Diamond, who is of course a neuro-oncologist, so he's really a specialist at this area. And I just want to point out that actual infiltration of MAP kinase pathway activated cells in the brain parenchyma is actually very rare, but there are, in quite a substantial number of patients, you do find granulomatous or tumorous lesions in the brain or in the spinal cord, and also at non-parenchymal sites like the base of the skull or the dura. And this is a very good example in a really nice overview paper written by Fleur Cohen-Obar together with Eli Diamond. So if you want to know more about the neurodegeneration and central nervous system histiocytosis, please look at this really nice paper. So when you look at the lesions in the brain, they actually biologically and histologically look the same as other LCH lesions anywhere else in the body. So they will contain cells expressing CD1A and CD207. And very importantly to notice that up to 25% of all patients that present with some form of Langerhans cell histiocytosis will have an affected HPA axis, and the most common form is diabetes insipidus. So then the real neurodegeneration, it's a topic that has been discussed earlier during this meeting, and I think it will come back for many, many more times because it is really an important issue to address. So it's really biologically distinct from the tumorous lesions. It can take some time before the neurodegeneration becomes clinically apparent. It can take years after the primary diagnosis of LCH. Is a very low incidence, fortunately. That's a paper published by the French group. But what is known is that even if you do not have real clinical signs of neurodegeneration, you still can have abnormal CNS imaging. And this is a clear example also from the paper by Fleur and by Eli, showing us cerebellar atrophy in a clear case of neurodegenerative LCH. So this is a treatment algorithm that Eli discussed with us. So it really depends on whether the central nervous system involvement is a single site presentation or multi-site presentation, whether or not there are already signs of neurodegeneration at presentation. And here you can see the different algorithms that you can choose from in terms of whether you want to sit and wait or whether you want to actively treat these patients, for instance, with the inhibitors, although we heard already on Sunday that once the neurodegeneration has kicked in, the inhibitors will not induce major, in most of the patients, the inhibitors do not induce major improvement. So again, please go and check this paper if you want to know more about this. What I personally like very much is that there's a category of patients that is intolerant to these MAP kinase pathway inhibitors, or there is certain comorbidities which does not allow these inhibitors to be used. And this is work published by Jasmine Francis, and she's actually here in the meeting this, so I don't know, Jasmine, are you sitting? I can't see. But they tried in 14 patients intraarterial melphalan treatments. These were all adults and adolescents, and they got really impressive results of regression of lesions, for instance, in the orbital area, but also in the maxillary sinus by this intraarterial melphalan. So I think this is, for those patients that do not qualify for the inhibitor therapy, could be an interesting alternative. We also invited Marita Partane. She's a neuropsychologist from Canada, and she discussed with us available literature and what is known about neuropsychological outcomes in Langerhans cell histiocytosis. And it turns out that there is really not a lot of information on this. I think, Fazanta, you are the one that is really here, that has done a lot of work in this area. So please discuss with Fazanta if you are interested in this. This is data from Fazanta published in the JCO already quite a number of years ago. And what you can see, this is the comparison of different types of IQ testing, 3 different forms, and then she compared 3 groups of patients with Langerhans cell histiocytosis with no central nervous system involvement, patients with only DI, and patients with CNS involvement, either only abnormal imaging results or already patients with neurodegenerative LCH. And you can clearly see that also in terms of neuropsychological evaluation, these patients do not perform very well. And this is an overview of all the cognitive and behavioral studies in LCH, and you can see it is just a few studies with an enormous range of patients that have been investigated with different diagnoses and also only on a single time point. So I think Marita made it very clear that yes, we need to design a trial on inhibitors, that's very important, but also we need prospective longitudinal studies addressing neuropsychological issues in these patients. So this is also a call to you to really don't forget about this. And within the Prinses Maxima Institute in Utrecht, we are already thinking about a study in this direction. So another thing that we should not forget is the link between the neuroendocrine system and the immune system. Because the different systems really communicate very closely, and that is shown in this cartoon. So here is the HPA axis, and it produces all kinds of hormones, including glucocorticoids. And these hormones can actually also stimulate cells in the immune system, in the lymph nodes, in the spleen. If you have immune activation in these secondary lymphoid organs, there's a lot of production of cytokines that can lead to increased cytokine levels in the plasma, and that obviously can again affect processes that go on in the brain. And Peter Lehne discussed with us that it really depends on the level of glucocorticoids, what kind of responses are generated. So this is just to visualize that you can have a very limited production of glucocorticoids over a very short window of time. Then you will find monocyte influx in tissues. You will find pro-inflammatory, they will develop into pro-inflammatory monocytes producing all these inflammation-promoting cytokines. But this is really only a transient response. If there's prolonged periods of time where there's an elevated level of glucocorticoids in the plasma, you will have this will have a different impact on monocytes. So you will change their behavior. They can become phagocytic cells. They will produce all kinds of cytokines that also stimulate matrix deposition, like fibrosis, for instance. And you end up with macrophages producing different types of cytokines. So this just shows you that monocytes are really sentinels of stress, and depending on the level of glucocorticoids, you can have different types of monocyte activation. And then when we talk about cells that are in the brain that could be potentially affected by these kind of pro-inflammatory factors, there's this ongoing debate on the cell of origin. So we talk about BRAF-mutated, very, very early York sac-derived progenitors that may be seeded during embryonic life. They're seeded into various tissues. There they sit, and at some point in time, they can become active and then start maybe to cause this pathological responses. But there's also clearly data from the group of Carl that says no, if you have sufficient levels of circulating BRAF mutated cells in the circulation, there's actually a likelihood that these cells invade the brain parenchymal at some point in life and cause neurodegenerative LCH. So this is really a debate which is going on, and there's different experimental data in support of this origin, but there's also evidence in support of this hypothesis. So I'm not gonna make any arguments, but I really like the work that was discussed by Florent Ginoux from Singapore. And this picture was already shown yesterday, so that is quite nice for me. They use a very elegant reporter system because by doing transcriptomic analysis, they found out that in the mouse, when you look at the development of neutrophils and monocytes, that these are really the cells that come from a granulocyte monocyte precursor. And this particular precursor expresses a transcript of a MS4A3, and when you use a reporter system, you can actually trace on the level of the reporter, which is in this case a certain dye, tomato red. You can actually trace all the cells that are derived from this particular progenitor cell. So cells that come from the common dendritic cell progenitor, they will not stain red because they come from another progenitor. You can only see the offspring turned in red from this particular progenitor. So using this reporter system, Florent and his team actually addressed the contribution of bone marrow-derived monocytes in all these different tissues. So this is no inflammation, just steady state. The mice are born, you keep them alive, and you check in the organs at distinct ages, so up to 36 weeks of age. You can see that there's a real difference because the microglia cells in the brain, the epidermal Langerhans cells in the skin and the mucosal tissues, as well as the Kupfer cells in the liver, they are not replaced by cells that come from this bone marrow-derived progenitor. Opposite to the monocytes in the blood, obviously, they come from these progenitor cells, but also macrophages in the lungs and the spleen and in the peritoneum, as well as in the kidney. You see that at least part of these cells are replenished by monocytes, by macrophages derived from monocytes that have been generated from this progenitor cell. So he came up with this sort of hypothesis that there's certain types of monocytes, macrophage-like cells in the central nervous system, but they come from a different origin. So they're all long-lived. So we'll talk about chorioid plexus macrophages, meningeal macrophages, perivascular macrophages, and the microglia cells. They're all long-lived. And it seems that they are all from the yolk sac, so fetal origin, with one exception, which seems the chorioid plexus macrophages, because the chorioid plexus really forms the interface between the blood and the brain. So it seems that at least part of these macrophages can be derived from a monocyte or blood-derived progenitor cell. And the question really is this a monocyte derived from the blood, or is this a monocyte derived from sort of local monocyte depository? Because other data published in Science last year shows that at the CNS borders, there's a substantial pool of monocytes that resides there that's not blood-derived. It just sits there, but when there's some kind of injury inflicted to the central nervous system, it's cells that are recruited from this local pool that then invade the brain for maybe induced tissue regeneration or at least solve the problems which have occurred. And then we come to the data that were discussed by Jennifer Pekarsik. This is work that she did together with Ken McLean. And Jennifer clearly showed that indeed in an autopsied brain material from a patient who died from neurodegenerative LCH, you do find monocytic-like cells expressing the mutant BRAF V600E protein depicted here by the VE1 positive VE1 staining, myeloid staining, CD33 positive cells. The cells also express CD14, and importantly, they also express the CD163 marker. And again, in that same regions in the brain where you see these perivascular macrophages, but also this tissue-invading macrophage, monocyte macrophage-like cells, you see the production of MCP1, which is a chemokine that attracts monocytes. So this is the issue. We're talking about microglia cells, which at least Florent and others claim that they come from the yolk sac, from a progenitor that comes from the yolk sac. They're seeded and they stay in the brain for a very long time, versus the monocytes that are derived from progenitors in the bone marrow that can also migrate to tissues, but they have a different expression of markers. And interesting, you look at this marker, which is a real exclusive marker for the microglia cells. And when you look in the autopsy brain material, there are some cells, some microglia cells expressing these markers, but it is a very, very, very low number as compared to the cells that stain with the VE1 antibody. So it really seems that in this autopsy brain, the cells come from somewhere else and at least are not tissue-resident macrophages. Ergler was the second Pritchard Fellow that discussed with us her work about dendritic cell ontology, and especially the type of cells that seem to be the immediate precursors of the neoplastic cells that we find in LCH lesions. So I'm not going to discuss it in detail because she gave a very nice presentation yesterday. I just want to point out one thing, and that's the DC3 cell, which carries this CD163 marker. And I think that Jen has been looking at these DC3 type Glycol cells that for some reason in the brain might just develop more into monocyte macrophage. And in some other occasions you will have a differentiation of these cells in more Langerhans cell-like cells expressing the classical CD1A and the CD207. And I don't think this debate is ending, but I think we are now really getting more pieces of the puzzle to really find out how these neoplastic Langerhans cells are actually generated. And then it's also interesting to point out that there's more and more evidence that there is some form of overlapping myeloid spectrum in all these different histiocytic disease entities. So the LCH, the RDD, and the xanthogranulomas family, they all have these monocyte macrophage-like cells, and it just, to me, it tells you there's some kind of plasticity in these precursor cells, because depending on the situation where the cells end up, that will determine their ultimate phenotype. Matthias also came to, was the second Pritchard Fellow that came to Athens, and he already discussed his beautiful work in the mouse model and the comparison with the human with the human brains, sorry. And it's interesting to see that both in mice in this transgenic mouse model, and there's a lot of issues with this mouse model, but if we put that all aside, it's interesting to see that the cerebellum, both in mice and men, seems to be affected. And again, that really all seems to be related to the neurodegenerative LCH. And what I personally liked very much is, and he didn't show it yesterday, so that's why I think it's nice to show. He really did a lot of different functional testing, and yesterday he discussed the data with the box. And this is also nice, the grip strength setup. So you ask the mice to pull itself up, and you can measure its strength. And it really seems that the mice that express these mutant BRAF cells, that they lost part of their grip strength. So it's not only the accumulation of these cells in these different areas of the brain, it's also loss of motor function. So I think that is really interesting data that come out from this mouse model. So the last part of the presentation is about the talks about novel therapies that fight central nervous system disease, and we invited people that are very active in the Alzheimer's disease community. And it's important to realize that this choroid plexus, which really forms the border between the blood vessels and the brain parenchymal, and you see these perivascular macrophages that are here at the basal membrane layer that separates the blood from the brain tissue. These really seem an important population that may be important for the regulation of the influx of polymorphic neutrophils in response to bacterial viral infections, but also, for instance, in the context of Alzheimer's disease, where you need to get rid of these amyloid beta peptides and these plaques that are formed, and they can really maybe play a very important role. So Kate Monroe discussed with us very interesting work in how to target cells that live in the brain. As you know, the brain is not very easy penetrable, so if you want to have an antibody therapy or whatever, you need to come up with a clever way to get this antibody across the blood-brain barrier. So they focus on activating antibodies that bind to the TREM2 receptor. And the TREM2 receptor is an important receptor expressed by microglia cells, and it is important for their function. So here's what happens. Normally these TREM2 receptors bind lipid ligands, and you get an intracellular activation, a little bit like the MAP kinase pathway, but then a different pathway. And this will, if for some reason, if the TREM2 receptor is not activated, you have loss of phagocytic function, the cells die, and they get into this pathological state. So they made a fully humanized antibody which with the FAP2 fragments recognize the TREM2 receptor, but on the FC portion of the antibody, they modified this so this antibody has a very high avidity for the transferrin receptor. And that's interesting because this transferrin receptor is actually expressed by these endothelial cells that form the border between the blood and the brain. So here you can see the antibody uptake by this transferrin receptor, and it's then released into the brain parenchymal. And as you can see here, this is just the antibody concentration in the brain measured after a few, I think, 5 different intravenous injections, and you can see that it really makes a difference if you modify the FC tail of this antibody and make sure that it is now able to bind with a very high avidity to this receptor. So that is another way to replenish what is lost because of pathology, is by generating new cells and by using induced pluripotent stem cells. And Matthew Blurton-Jones, he is from the US. He has a lot of experience in generating monocytes, macrophage-like cells, but also microglia cells using PBMCs from patients with Alzheimer's disease. And he can make these cells in vitro, and then he can inject them into the brain. And this is just pictures, and he uses a specific mouse strain which does not express the mouse MCSF receptor, but it expresses the human MCSF receptor. So these mice are completely devoid of Langerhans cells and microglia cells, as you can see here, because these are the control mice that received PBS. So you see in green staining the microglia cells, so you see there's hardly no cells. But if he injects these iPS-derived microglia cells, this is a picture about 4 to 5 weeks after injection of these cells, and you see they migrate to all these different areas in the brain, and they sit there. And not only they sit there, they also seem functional, because if you look at their transcriptomic activity, and here they are depicted in purple, they seem to have the same transcriptomic activity as compared to microglia cells taken from autopsied human brain. So they not only sit there and survive, but they also seem to be active. So I think this is very, very promising for, well, if you have, for instance, microglia cell loss, that this could be an alternative way of replenish these cells. And then finally, we had the pleasure of inviting Don Cleveland. Don is actually, as he told me over dinner, he is one of the inventors of the tau protein. So he has been working in the Alzheimer's disease community for a very, very long time. And he and his team explores therapeutic potential of antisense oligonucleotides. And this is what normally happens with gene transcription. So there's a messenger RNA formed after transcribing the DNA, and that messenger RNA is released and then translated into proteins. And then if you can have a clever design, you can make an antisense oligonucleotide that binds to this messenger RNA molecule, and thereby you can prevent the translation of this RNA into protein. So this is a technique that has been around for a long time, but I think it's very clever how he explored it, because they target PTP1, and PTP1 is a very important transcription factor for the generation of neurons. I'm finished in two minutes. So here you can see the level of PTP1 messenger RNA in the brain just a few days after injection, intra-brain injection. You can see there's a drop in the hippocampus. It's not 0, but at least it's diminished in the motor cortex, but a little bit less in different parts of the brain. It's easily recovered within a few days after injection. But when you look at the brain in the different parts, so in the hippocampus, but also in the cortex, you can see, again, using this tomato reporter system, you can see these very beautiful neurons with all these very nice dendrites. And also these cells seem to be present for a long time, and they seem transcriptionally active. So I think in the meeting we had really a lot of nice discussions about how to deal with these kind of diseases, how to replenish the cells. So obviously we are not there yet to really come with a solution for neurodegenerative LCH, but at least we got some nice clues in what kind of areas we should think about. Just to make it a little bit more complex, and this is my last slide. The brain is a very complex organ, and here you can see the transcriptomic activity of all the different regions in the brain with a very nice technique, Murfish, if you are interested. So these are different probes, and then you can see all the different types of CNS cells with their own transcriptional activity. And that's it. So, on behalf of Paul, Elizabeth, and Nick, yeah, I hope you enjoyed this talk. I'm open for questions, but maybe you can help me.
No, first, Astrid, thank you. That was absolutely fantastic, because it is very difficult to pull together all these different talks, and it's not your data, so you did a fantastic job, a really clear message. Are there any questions from the audience? And I'm aware that there's an online community, so I don't see any questions there. Okay, well,
There's one.
Oh, sorry, yeah, okay.
Astrid, thank you for a good overview, but can I just go back to the CNS involvement with LCH? And Blaise Favara always used to say that when he was doing pathology of the patients with LCH in the brain, that you didn't get CD1A expression in the actual infiltrative areas, but you do get CD1A when you have infiltration from bone lesions.
Yeah.
So how much is infiltration from bone lesions versus infiltration, perivascular infiltration?
Jennifer, where are you?
She was
Jen is
sitting
not
over
in?
there.
Okay, so I think you're absolutely right. It really depends on where the lesion originally started. So if it's a bone lesion, maybe there is more CD1A, CD207, because you have bone-morphic proteins, TGF-beta, all kinds of factors produced in the bone that may stimulate the expression of the CD207 and the CD1A. And maybe with these kind of lesions, because they originate from different cells, different microenvironments, and that's maybe the reason why you end up with a more monocytic macrophage-like cell type. I published myself, you can get these progenitors, you can grow them in different directions, dendritic cells, Langerhans cells, monocytes. You all end up with cells expressing the BRAF mutation, but just a different phenotype, simply because we throw in different growth factors. So I don't think it will tell us that we can make a strict separation between cells in the brain being more macrophage-like cells, that they come from a different cell. I don't think we have enough data to really draw that conclusion. But that's my opinion. Maybe other people think differently.
Okay. Thanks. Just one quick announcement, actually. Astrid mentioned the Pritchard Scholars, and any young investigators, it's a great opportunity to come and present your work and interact with a really top team of scientists. So that will be announced in the spring, so do look out for that. Everything's paid for, and it's great fun, as you can see. So I'd just like to thank you again for a fantastic presentation, and we can continue over coffee.
Yeah.